Okay, so let's start. So hello, everybody. Welcome to the AES ESA inaugural talk. I'm Jiafu Mao from Oak Ridge National Laboratory. Yeah. And I'm the current chair of the Asian Ecology section of the Ecological Society of America. Established in 1994, AES is deeply committed to supporting ecologists who are advancing groundbreaking ecological research across Asia. Aligned with the Ecological Society of America, our shared goal is to enhance communication and foster collaboration among ecologists who focus on the rich diversity of Asian ecosystems. Together, we champion critical ecological research within Asia and also serve as a vital nexus connecting researchers, societies, and groups involved in diverse ecological disciplines within the region. So currently, AES is proud to have 5 executive members serving from August 2023 to August 2025. Our executive committee members include myself, Vice Chair Professor Lei-Xin Wang from Indiana University, Indianapolis, Secretary and Speaker Professor Wei Ren from the University of Connecticut, our student Liaison Wasabi Prakash, a Ph.D. student from Auburn University, and our former chair, Professor Dafeng Hui from Tennessee State University. So please refer to our website for more detailed information about the Asian Ecology section of the Ecological Society of America. So to keep our members informed about pioneering research and to encourage collaboration, we are delighted to launch our virtual AES ESA Talks series, occurring approximately every 3 months. The series will feature a wide array of speakers, ranging from our own members to representatives from prestigious organizations and leading project directors. So tonight, we are excited to host Professor Hojeong Kang from Yonsei University, Seoul, Korea, for our inaugural AES ESA talk. Professor Kang will present on soil microbials and drivers of the global carbon cycle. So Professor Kang holds the position of full professor at the School of Civil and Environmental Engineering, Yonsei University, Seoul, Korea. His primary research area centers on the impacts of global climate change on microbial ecology, particularly in relation to greenhouse gas emissions. Professor Kang earned his bachelor's and master's degrees from Seoul National University before completing his Ph.D. at the University of Wales, Bangor, UK. His career trajectory includes postdoctoral work at the University of Wisconsin-Madison, USA, and a professorship at Ewha Womans University, Seoul, Korea. Professor Kang has authored and co-authored more than 140 papers in international journals, including 4 publications in Nature research journals. Currently, he also holds the role of an associate editor for Ecological Engineering. Now let's welcome Professor Kang for our first AES ESA seminar.
Hey, thank you very much, Jeff, for the nice introduction. Let me share my slide first, and then going to start my talk. Okay, can you see my slide and hear me clearly?
Yeah, clear.
Okay. Right. Good evening in the States and good morning in Beijing, Seoul, and probably in Tokyo and other parts in Asia. My name is Hojeong Kang from Yonsei University, Seoul, Korea. And I'm very pleased and honored to have an opportunity to share my research with you. And particularly, I'd like to thank you all for joining my talk. And I appreciate the AES ESA committee member for inviting me to this talk. So the topic that I'd like to address today is microbes as a driver of carbon cycle. Firstly, I'd like to acknowledge my co-authors and collaborators, including Professor Freeman in Bangor University and Professor Sting and Yuan in Institute of Soil Science, and Professor Rushford in Laval University and Professor Zhuang in Purdue University, and my current and former members at Yonsei University. So I'd like to thank National Research Foundation and Ministry of Environment for financial support as well. So firstly, I'd like to give you a background information about my research in connection with the climate change and carbon cycle and microbes. And then I'd like to showcase 3 research examples, including peatland carbon storage, invasive plant effects on methane emission in a coastal wetland, and methane oxidation in forest soils. And then I'm going to round up the talk by making several conclusions and propose some outlook in this field. So, the temperature is continuously soaring up, and we are experiencing a drought in everywhere. Also, this is about sea level rising, and which affect coastal wetland substantially. And increasing carbon dioxide concentration itself could be another impact on terrestrial ecosystems. All these impacts are caused by carbon dioxide released by human activity, of which is estimated up to 11 petagram per year. And about half of them are accumulating in the atmosphere, causing all this kind of climate change issues. However, as an ecologist, you guys are all aware of the much greater cycle behind it. For example, about over 120 petagram of carbon are observed by photosynthesis, same in the ocean, and about half of them are going back to the air by heterotrophic respiration. Especially my research focuses on the carbon release from the soil, which is one of the biggest carbon stock on Earth. And this is mainly about carbon dioxide. And also there is a huge flux of methane as well. And the methanes mainly come from agriculture, rice paddies or cattle. Also, they are released in large amount from wetland and other aquatic systems. But at the same time, about up to 5% of methane in the atmosphere is removed by oxidation of the group of bacteria named the methanotropes, which draw much attention as well. So when we are looking at a carbon cycle in ecosystems, many ecologists are looking at the amount of carbon stored in biomass and soil organic matter, as well as they are looking at the amount of CO2 absorbed by photosynthesis and released by respiration. And some of carbon added into the soil is in the form of litter, and some of the organic matter are decomposed and released back to the atmosphere, either as a carbon dioxide or methane, depending on the redox potential of the system. And some of them are released to the adjacent aquatic ecosystem as a dissolved organic carbon. And in aerobic condition, some methane can be oxidized, turning into carbon dioxide as well. Among these carbon cycle processes, the decomposition of soil organic matter is mainly mediated by microbes, but compared with other components of carbon cycles, relatively less information is available, and the mechanism has not been revealed yet. So having said microbes, as the name implies, microbes are very small in size, but they are great in their numbers and diversity. But the difficulty is that majority of microbes are not culturable. We cannot grow them in the lab. So these days, what we do is we are collecting samples from the field and extract their nucleic acid and sequence it, or sometimes analyze in different ways, and then collect the information, and then try to understand their abundances as well as diversity and community composition. So in my lab, we use the conventional Illumina MiSeq or real-time qPCR to better understand microbial community structure and abundances. We also run stable isotope probing or mRNA-based methodology to tease apart active microbes only. And with those information, we try to connect them to process rates of the soil system by analyzing enzyme activities and trace gas emissions. So with that background, I'd like to introduce a couple of my previous research. And the first one is about peatland and carbon storage in those ecosystems. Peatland is a place filled with peat, undecomposed organic matter, and they are widely distributed in far northern areas such as Alaska, Canada, and northern Europe and Siberia. Also, some of them are found in tropical regions such as Malaysia and Indonesia, and some part in Africa. And the importance of this ecosystem is that they are estimated to store up to 1,000 petagram of carbon. And the question is why decomposition is so slow in this peatland. In long time ago, our group proposed so-called enzyme latch hypothesis, where the peat soil is flooded with water, so oxygen level is very low. And the system is enriched with amorphous chemicals named phenolics, which are highly recalcitrant, as well as inhibitory against microbial activities. But if the water level draws down, the oxygen penetrates, and this is known to activate an enzyme named phenol oxidase, and then which eventually decompose phenolics, and then the inhibitory effects disappear, and consequently microbial activities are enhanced and release carbon dioxide and methane and DOC. And more recently, we have shown that pH is another important controlling variable for phenol oxidase. In this graph in the left panel is the data from global survey of phenol oxidase in relation to pH. And these data were collected in the UK, Korea, Japan, and the Kalimantan and tropical regions. And as you can see here, there is a strong positive correlation between pH and phenol oxidase. On the right panel, this is a manipulation experiment where we added alkaline or acid over 2 years in peatland, so intentionally changing the pH in the system. Again, we saw very strong positive correlation between pH and phenol oxidase, and both data indicating that pH is another important controlling variables for phenol oxidase, and consequently the composition of peat matrix. And then how this kind of information can be applied in terms of carbon storage and restoration of peatland. For example, this is photos taken in Canada, where they dug up the peat to use it and drain the system. And as I mentioned in previous slides, this drawdown of water level will activate phenol oxidase and eventually decompose the peat in deep soil and may release more carbon. So they want to restore this system and maximize primary productivity, as well as slow down the decomposition so that we can store more carbon in the peatland. How can we do that? Firstly, as the enzyme-ledge hypothesis suggested, simply by rewetting the peatland can reduce oxygen concentration in the system, and then slow down the phenol oxidase activities, and then store more carbon in the system. Or we can artificially apply the phenolics in the system as a form of some debris from timber industry, or even biochar or other materials which may have a high concentration of phenolics, and this can latch the phenol oxidase activity as well, and can slow down the decomposition. Finally, if we can plant sphagnum and successfully restore the sphagnum peatland, we could slow down decomposition as well, because sphagnum release various organic acid and can lower the pH of the peatland systems, so that this can further slow down the phenol oxidase, and we could stabilize and store more carbon in the system. So now we are working on this hypothesis and implication at the field scale in Canada. And the second set is about coastal wetland, especially effects of invasive plant on methane emission. This is a photo taken in east coast of the United States. The area was originally dominated by Spartina alterniflora, which is a native species in North America. But the area is invaded by Phragmites australis, which originated from Eurasia, and they are invading many parts of the coast of the United States. And interestingly, the invasion is actually the other way around. In United States, the coastline is invaded by Phragmites australis, originated from Eurasia. The east coast of China and many parts in South Korea are invaded by Spartina alterniflora, or sometimes Spartina anglica, originated from North America or Europe, respectively. So we were interested in the impacts of such invasion on methane emission from the site. And many previous studies have documented the effects of such invasion on plant biodiversity, or invertebrates and other components of ecosystems. But less studies have been conducted in relation to methane. And one of our previous study on the left panel, we compare the methane fluxes in different vegetation type in east coast of China. This graph is the methane emission from the Phragmites australis dominated site, so native plant. However, if the site is invaded by Spartina autumniflora, as you can see here, more methane comes out from the invaded site. And we did a meta-analysis from published data as well, and we successfully compiled over 270 comparative studies where they measured methane emission between native species and places with invaded by invasive species. And again, there was a statistically significant differences between the two. So why invasion of the plants may increase methane emission from the site? Basically, invasive species grow bigger and faster, so they provide more carbon to the soil to feed microbes and especially methanogens, which produce methane. Also, because their biomass is big, they function as a conduit of gas passage, which further accelerate methane emission. But we didn't have very direct evidence in terms of microbes. So we conducted some experiment in the measurement in Korea. This is important because, as you can see in the left panel, the methane concentration increasing continuously, but still methane budget is yet to be closed compared with the carbon dioxide. And methane dynamics is very complicated in the wetland system as well. Basically, the organic carbon in the system is first fermented to hydrogen, carbon dioxide, and acetate, and then they are metabolized by methanogens and then released to the atmosphere through a diffusion, evolution, or sometimes through the plant biomass. And the methanogens, to detect methanogens, we are targeting enzyme, the gene, MCRAA. But some of the methane produced in the system can also be oxidized by other group of bacteria named methanotropes, where the bacteria can oxidize methane, convert it to carbon dioxide. And their main gene responsible this process is PMOA gene. So by assessing these gene abundances and community structures, we could better understand the mechanism and dynamics behind the effects of invasive plant. So to address this issue, we conducted a study in Suncheon Bay, the southern part of Korea. The site is originally dominated by Sueda japonica in the mudflat setting. But in last decades, the area is invaded by Phragmites rapidly. Actually, Phragmites is not exotic species here, but they are invasive. They are expanding their habitat very rapidly. So by comparing methane emission and microbes in these two contrasting systems, we aim to better understand what's the behind the mechanism for that. So first, we measure methane fluxes, and as you can see here, this closed circle represent a methane emission from a Phragmites site, the invaded site, and this open bar is the methane emission from Sueda, the native species. So clearly show the same trend as reported in other studies. We also measured methane production potential. It's basically we collect the soil and incubate them under optimal condition for methane production. So we make the system anaerobic. We added extra carbon and incubated in warm temperature. And we did it at different depth of soil. And black bar represent the Phragmites site, and the gray bar is a Sueda site. And as you can see here in the deep soil, the methane production potential is significantly higher in the phragmite site. And what's the mechanism behind it? First, in the graph, the upper panel is phragmite site again, and lower panel is Sueda. And this is a depth profile from the surface up to 1 meter. And different color represent a different sampling period. And as you can see here in green graph, the dissolved organic carbon concentration in phragmite site is significantly higher, especially during the summer season. So when the plant grow actively, they provide extra carbon into the below ground. At the same time, the water content in the soil of phragmite site is higher than Sueda site. That's quite unexpected result because in terms of the location, Sueda is usually developed close to the seawater, so they are more frequently flooded. However, in phragmite site, because the biomass is so big, they've got a stronger shading effect, so they just cover the sunlight to reduce the evaporation. So in the soil system, actually, phragmite site has got a higher water content, so wetter and lower oxygen concentration, so consequently better condition for methane production and emission. And finally, we look into microbial abundances of methanogen and methanotroph, and we calculate out the ratio of the two. So if this number is high, that means the condition is good, better for methane production. And if the number is low, the system is less methane production and more methane oxidation. And I'd like to draw your attention in this part, in between 50 to 80 centimeter from the surface. The ratio in the phragmite site is significantly greater than Sueda site, indicating that even microbial abundances in the phragmite site is better or formulate in a way that has got a higher methane production potential. So based on this information and other microbial information, we propose the microbial mechanism for methane emission by invasive plant in this cartoon. So if the site is invaded by invasive plant or exotic species, usually it has got a higher primary production and more root exudates and plant debris. So more carbon are supplied to the soil, and this will eventually increase methanogenesis. And at the same time, the methane oxidation are relatively smaller in those systems. And also, I didn't show in this presentation, but we saw a reduction in sulfate reduction, and in combinedly, this result in greater emission of methane from the invaded site. Finally, I'd like to shift gear and move on to aerobic condition of a forest. In the forest soil, the aerobic condition, there are bacteria named methanotrophs. And what they do is they oxidize methane and convert it to carbon dioxide to acquire energy for themselves. So this is a chemical reaction that are mediated by these methanotrophs. So they convert methane to carbon dioxide, but as you know well, methane is about 35 times stronger greenhouse gas than carbon dioxide on a molar basis. So this is good news for in terms of climate mitigation. And how can we measure it? We use a static chamber in the site. Basically, we install this closed chamber in the soil and then connect it to a mobile GC unit, and the gas is going into the machine and comes out. There is a circulation, and we note the changes in the gas concentration inside of this chamber, with which we can estimate methane oxidation rate. For example, here, the upper panel is the concentration of methane over the period of time. As you can see here, the concentration is decreasing continuously over a certain period of time. And by measuring the slope, we can estimate oxidation rate. And you also can see the increase in the carbon dioxide concentration. And this CO2 mainly comes from the heterotrophic respiration in the soil. So we monitored the methane oxidation rate in forest in South Korea in 2 locations. One in Gwangnam Forest near Seoul, and the other one is a Hannam Forest in Jeju Island in South Korea. We have 2 different objectives of research. In the Gwangnam Forest, we compare the methane oxidation rate in forest with different thinning practice. Thinning is one of the forest management scheme where instead of maintaining natural forests, sometimes they cut down the trees to reduce the density of forest to increase actually timber production. So if the number is decreased, density decreased, actually single trees grow bigger and thicker. So we measured methane oxidation rates in the forest with the same species but different thinning intensity. In Jeju Island, we measure methane oxidation rates in different vegetation type, the deciduous forest versus a coniferous, and also a mixed forest as well, to understand the effects of tree species on methane oxidation rate. So we measured these rates over a 2 years period, and there is a seasonal fluctuation, and there are 4 different colors. This brown color is control, so we did not do any thinning, and the density is about 800 trees per hectare, and light thinning, moderate thinning, and heavy thinning. And so we compare methane oxidation rates in different thinning intensity, and we saw the highest methane oxidation rate in moderate intensity of thinning compared with control or heavy thinning plots. Regarding tree species, we observed a significantly greater methane oxidation rate in oak, the deciduous tree, compared with Japanese cedar, which is coniferous. So broadleaf forest showed a greater methane oxidation rate than the coniferous one. But a previous study have shown that probably temperature and rainfall events are or soil moisture would be the key controlling variable for methane oxidation, because that affects gas diffusivity and microbial activity in general. But in those sites, we did not see any significant correlation between methane oxidation rates and temperature or water content. Instead, we saw a significant positive correlation between soil organic carbon and methane oxidation rates. This is quite an unexpected result, because general belief so far is that if there are more soil organic matter, then probably that kind of help the heterotrophs to dominate, or if the condition is wet enough, methanogenesis can occur. Methane oxidizers are autotroph. So if the heterotroph are dominant, probably they may lose their competitiveness and their activity might be small. So in overall, if the soil organic matter are high, that may reduce soil methane uptake and oxidation rate. This is well documented in many studies previously, but our result shows a different trend. So to confirm our finding, it can be generalized. We collected data from published literature. We successfully found 81 papers with methane oxidation rates reported, as well as the soil organic carbon from over 200 sites. And we put all the data into one graph. Again, there is a huge variation, of course, because the data collected from all biomes, but still there is a significant positive correlation between soil organic carbon and methane uptake rate, suggesting that our finding in Korea was not, peculiar one, but it can be generalized in some way. If we divide organic matter in several brackets, we saw the same trend. And so far, the best estimate on methane oxidation rates globally is based on the paper by Liu et al. 2020, where they estimate methane oxidation rate is a function of methane concentration in the atmosphere, soil temperature, soil moisture, and soil redox potential. But our data suggests that probably soil organic carbon is another important factor. So we included the function of soil organic carbon in the model, and we tested the model accuracy as well as a global estimate of methane oxidation and forest. First, in the left panel is the previous model, and right panel is the model incorporating soil organic carbon. And our model has got a lower RMSE, indicating that our model is more accurate in terms of the estimation of methane oxidation in soil. And then we apply that model to a global scale. In the left panel is the previous model estimate, and right is a new model based on ours. And we conclude that actually the methane oxidation in global forest is about 40% greater than previous estimate. So these are the result of mine. So the takeaways of my talk is the first, soil ecosystems play a key role in global carbon cycle. And microbial information is essential to elucidate and understand the mechanism behind it. And the thing is that this climate change, the environmental change affect microbes, but at the same time, as you've seen in this talk, microbes can increase or decrease carbon dioxide, methane emission from the ecosystems. So there is another biological effect from them. So this so-called biological feedback need warrant further investigation. Due to time constraint, I present just 3 sets of data, but currently I'm looking at the warming effects on microbes in Arctic soils as well. And also we are looking at methane oxidation in arid and semi-arid ecosystem, which have not been explored. We're looking at that aspect as well. And finally, we are interested in so-called blue carbon. And particularly, I'm interested in why organic matter decomposition is so slow in mangroves and other coastal wetland. These topics are I'm working on now. I'm more than happy to discuss about them more in other chances, or to find collaborators both in the States and China or any other Asian countries. With that, I'd like to stop here, and I'd like to thank my lab members again. And if you got questions, comments, you are very welcome to email me at hj_kang@yonsei.ac.kr. And also, you may want to explore my website, eel.yonsei.ac.kr. Most of the information in English, so you may want to explore more with that. Again, thanks for your attention, and I would be happy to entertain any questions you may have. Thank you.
Great. Yes, thank you, Professor Kang, for your excellent presentation. And now let's take the questions from our audience. Yeah, you can just either raise your hand or you leave your questions in the chat. So yeah, Li Xin, go ahead if you have a question.
Oh, sorry. I'm just clapping. I'm not asking a question. I could. Dr. Kang, yeah, thank you so much for the interesting talk. I'm more, a water person. So I'm just curious for the, when you talk about the forest oxidation or like uptake. So I guess my understanding is that if you have a more organic carbon, you could have both like a higher rate of oxidation and also higher rate of uptake. So I guess must be a balance when you, I guess, just try to get your thoughts about that.
Right. Yeah. So somehow there's a echoing. Okay. So the novel point in our paper is that previous studies suggest that temperature and water content are the main key controlling variables, because temperature, obviously all the biological activities are based on the temperature, and the water content, because if the soil is flooded or wet, gas diffusivity decrease, so less chance for methanotrophs to oxidize methane. But so we expect that kind of trend in Korea, but the temperature and the soil moisture appears to be not key controlling variables here, but organic matter. But actually, if there are more organic matter, that probably the bulk density would be lower. So probably more gas can be diffused into the system. And in previous models, they just assume negative correlation between water content and methane oxidation rate. But I suppose it's not just a simple negative correlation. But if soil is too dry, that may inhibit microbial activity in general. So up until certain moisture content, the actually methane oxidation goes up. But if that is flooded with water, like a heavy rainfall event or something like that, then that definitely decrease the oxidation rate. So that's the reason why my field data didn't show any single relationship between. So both oxygen level or temperature effect or bulk density and everything can be presented by a simple index of soil organic carbon. That's main argument of my paper here.
Thanks. Yeah, I'm interested in dryland. Particularly, I saw you have a research interest for like a methane oxidation or methane dynamics in dryland. I assume you are talking about for dryland, it's only after rainfall events you have a pause of CO2 or like other fluxes. I assume mostly you are focused on that type of pause response after rainfall events. Is that correct?
Yeah, that's a great question. Actually, I have currently very limited data on it, but my kind of hypothesis is that currently, they assume methane oxidation is zero in arid or semi-arid area. But I did a small project in Tunisia and other part of dry land, and there is many parts covered with the so-called biological soil crust. So even if arid and semi-arid, many regions covered with the thin layer of microbes, and actually they shows quite high activities of other things. And another thing is that because methanotrophs are kind of very slow growing bacteria, but they are autotroph. So if it's extremely drought condition, typical heterotrophs cannot endure that condition. So it's like a Martian environment.
Right.
And if there be any microbes, I kind of speculate that the autotrophs that can metabolize one carbon in a compound probably would be there. So, but there is a very few literature and nor actual measurement. So I did a simple project in Mongolia, and we have some evidence that actually methane oxidation can occur in dry land. But at this moment, I'd like to expand the measurement in other dry land, and hopefully enough data is accumulated, then we can modify the model to include a methane oxidation rate in arid or semi-arid land as well. But it's still a long way to go, and hopefully.
So sorry, just one last quick question. So for methane, we know for CO2, we know there is a linear increase, right? So I know methane is more complex. We know like the global level, there is a kind of a level up between 2000 and 2010.
Yeah.
We really have a good understanding what exactly is the cause of that level up.
Yeah, that's a great question, but there's, as I understand, there's a lot of scientists with different perspectives try to explain it, and there's a atmospheric chemistry groups who argue that hiatus in early 2000 to 2006 is due to the hydroxyl radical, because about 90% of methane can be oxidized by hydroxyl radical in the atmosphere. So they strongly argue that oxidation is enhanced during that period, and that can be explained by the thing. But other groups of people argue that probably the methane emission from a natural wetland is slowed down during that period due to global scale drought, and then there's the wetland areas wet again, so it's soared up. And the paper appeared in Nature, I think, this year from the scientists in Beijing. They show that the surge of methane from in last year, even during the COVID era, is mainly half can be explained by wetland, but half explained by reduction is air pollutant, and eventually that decrease hydroxyl radicals in the atmosphere. So I think the discussion is still on the way, and I try to understand and explain that by my own perspective. That is probably methane emission from wetland could be an good explanation for that. But again, it's still, I think, long way to go. Right. And my data is mainly based on site scale measurement, and I'm not a modeler, but I'd like to collaborate with modelers in the future to have a more better picture and view on that issue.
All right. Yeah. Thank you so much for the interesting talk and for answering my questions. I guess.
Thank you.
Dafeng has a hands up too.
Good. I see Dafeng has a question.
Dr. Excellent talk. Very interesting. I have a follow-up question. I see you made both the CO2 and the CH4 here. So did you analyze the CO2 data? Do you see the relationship between the CO2 emission and the temperature and the moisture at the site here?
Oh yeah, CO2 definitely shows a very strong, effects of temperature. So, methane is always a problem. CO2 shows a very nice, documented, Q10 value kind of thing. So, and the fluctuation is less dramatic. So I think both field measurement and model quite well, explain and present the carbon dioxide emission from the forest soil or other types of soil systems. But methane is very problematic, especially methane oxidation is less, variation, but wetland system. First, the production is very, unpredictable. I mean, depending on the system, and the way that released to the atmosphere is very diverse as well. As I mentioned before, diffusion, evolution, and even through the plant biomass. So if you look at the literature, the methane emission from aquatic ecosystem, depending on the paper, they pointed out very different, sources. Some say evolution is the dominant component, but others say, diffusion is component, main pathways or something like that. So not only the production or oxidation itself, but the way they release to the atmosphere is very diverse and hard to detect and measure. I think that's the kind of difficulties we are facing now. And I try to. So in your question, carbon dioxide is more straightforward and well fitted with the conventional models and understanding, but methane is still a long way to go to simulate.
Since you made both, at the site, reminded me of one study that Xiaofeng did quite some time ago. So basically, he kind of collected data from many sites of both the CO2 and the the CH4 emission, and then try to find a relationship. I think we found a linear relationship at many different sites and the systems. Yeah. So
all right. That's great.
Yeah. Did you try to see if this kind of CO2 and CH4, the uptake and the emission, could or not?
I haven't tried that yet, but yeah, that sounds very interesting. Yeah. I'll try it, and if I see any interesting trend, I'll let you know. Yeah.
Okay. Thank you. Very interesting talk.
Thank you. Thanks. Yeah, there is a question from Professor Hong. Yeah.
Hi, Ramsong speaking. Thank you for a wonderful presentation, Professor Kang. I have a question about your research related to Jeju Island. So in Jeju Island, even for oak species, now they are changing from deciduous oaks to evergreen oak species. So what species would be better for methane uptake? I mean, some say deciduous plants make small leaf biomass, but some say evergreen species has longer like growing season. So can you guess which species, like deciduous or broadleaf species, would be better for methane uptake?
Yeah, that's a great question. Actually, the background of the measurement in Jeju is related to shift in vegetation structure in that island due to the climate change. As the temperature goes up, many places the conifers are replaced by deciduous, and many models predict that trend as well. So my measurement is like a snapshot comparison, but the background is about that. And as Professor Song just pointed out, which species is better in terms of methane oxidation rate? And my conclusion is deciduous is better. And again, that provides more organic carbon to the system. But methane oxidation is just one aspect of the many ecosystem services that forests may provide. So if I just focus on methane oxidation, deciduous is better. But if you look at other things, I think there is an issue of biodiversity and, carbon storage in the soil or whatever, in many other things. So yeah, in terms of methane, deciduous is better, but you have to consider other ecosystems.
Right, thank you for the answer.
Propose and any suggestions to the policymaker or general public, I suppose.
I see. Thank you.
Thank you.
Dr. Kang, thank you. I have two questions. one more specific question about your invasive fragmenting, right?
Mhm.
You found that in the deep soil or 60 to 70 centimeter methane producing microbes to methane oxidizing, the microbe ratio is highest, right?
Yeah.
So do you have any idea what caused that?
Okay. Yeah. Our hypothesis that, you know, in the invasive site, they've got a deeper roots and more carbon available to the system. So, you know, if there are more carbon available, you know, more fermentation, more acetate, hydrogen, and carbon dioxide, so more substrate for methanogenesis. So that's the reason why methanogens are activated. But at the other the other way around, if the roots penetrate deeper, they may release more oxygen to the system. So some argue that may activate methane oxidation in the deep soil of the invaded site. So instead of just comparing methanogens or methanotropes separately, we calculate out the ratio. So if the methanogen over methanotrope ratio is high, then probably the system is more methane producing, you know, direction. And if the ratio is lower, it's more, you know, less methane production and more oxidation. So the main reason we believe is that first, more carbon supply. And secondly, as I shown before, fragmite site has got a higher water content, so more anaerobic system. So that's a better habitat for methanogens. That's our proposition.
Okay, thank you. And the more general question is that because invasive species usually have high biomass, say spartina, come into issue. So over the long term, do you think more invasive species will we have more carbon storage in soil?
Right.
And rather than, I mean, the number one thing for carbon sequestration is to stimulate the production, right? So
right.
how we balance these issues, or because we over the short term we measure that methane production or CO2 production, which actually is a tiny % of productivity. If we
Right. Yeah, that's a great question. Yeah, I just showed some methane emission in this talk, but we look at the carbon dioxide and nitrous oxide as well. And we calculate out, you know, total the CO2 equivalent budget. Actually, invasive plant store probably more carbon. So if we just focus on climate change issue, invasive species are not necessarily bad. But
okay,
as I mentioned in the previous question, there are many other diverse ecosystem services as well. For example, the mudflat site in Korea, if they are invaded by, you know, phragmites or other the Spartina anglica things, then that reduce the habitat for crab and other invertebrate and benthic creatures. So that actually reduce the biodiversity of animals, and then that could be another environmental disaster as well. So it's quite hard to tell which is the right decision, but in general, maybe invasive plants grow bigger and faster. They can sequester carbon in the short term, but there might be other side effect in terms of biodiversity, or even that can damage to the fishermen or local people. So yeah, it's very hard for me to which is the right way, but yeah, everything should be considered simultaneously to have a big picture.
Thank you very much.
Thank you.
Cool. So any more questions? Okay. If no, yeah. Thank you once again, Professor Kang, for your enlightening presentation. And also I would like to thank our audience for your engaging questions. So let's now bring today's AES ESA talk to a close. So we sincerely hope you will continue to join and support our future AES ESA talks, as well as other activities under the AES umbrella. Thank you. Thank you all.
Oh, thank you very much for having me again, and look forward to meeting you in person in some place in the future, probably in the AGU. Okay.
Yeah.
Thank you.
Everybody.
Bye.